Welcome to the SWAM course of Microbial Technology. Today we will be discussing about microbial diversity. In the previous topic, which was about organization of cell structure, we have seen that it was about cell, characteristics of cell, different types of cell based on their organization, their structure, that is prokaryote and eukaryote. Along with this, we saw about their examples with pictorial representation, their structures in detail and summary you all have seen. Moving on further, in today's topic we will be discussing about what is microbial diversity, their examples, some of their types, their application, their importance and summary. So, moving on to the introduction of microbial diversity. So, what is microbial diversity? We all know that there is certain variability among us, right? So, microbial diversity is defined as variability among living organisms. The main key of microbial diversity on earth is due to evolution. The structural and functional diversity of any cell represents its evolutionary event which occurred through Darwinian theory of natural selection. Microbiology and microbial diversity is the multitude of new unicellular organisms that can be outlined as microbial diversity. The cellular metabolism and transformation of these microbes are completely different. We all know that microorganisms are present everywhere. They thrive over the entire biosphere, defining the boundaries of life and allowing other living things to survive and evolve. The current diversity of microbes on earth is known and assumed to be very massive, but the exact extent of microbial diversity is still unclear. So, what do microbial diversity units actually look like? The answer is not exactly simple as it is for flora and fauna, though definitions keep on changing. Okay. So, we cannot completely rely on structural or physiological changes because under the microscope most of the microorganisms are indistinguishable and distantly related creatures can play comparable ecological roles. Microbes are able to swap genes across vast phylogenetic lengths. As a result, we rely on phylogenetic inference based on vertically inherited highly conserved genes. Microbial diversity is basically very much essential for human survival and economic security in the future, as it provides diverse range of resources that humans can use to their advantage. Understanding microbial diversity can aid in the mitigation of new threats such as diseases, pathogens and viruses. Microbes are also ubiquitous as you cannot avoid them. They live in and on almost everything, so they have to be significant. They have existed on this planet for much longer than we have, and they will most likely continue to do so in the future. Microbes can be found in places where humans have not ventured or are aware or are unaware where they actually exist. It is critical to understand how microbe diversity influences global genetic resource preservation and sustainability. As more extreme environments are explored, the richness of microbial diversity is becoming more apparent. Measures must be taken to estimate microbial diversity in order to maintain human health and nourish the human experience on a national and international level by wisely using and conserving genetic resources. Microbial diversity encompasses a vast array of microorganisms, each having unique characteristics and roles in various environments. The main types of microbes that contribute to microbial diversity include bacteria. So, bacteria we are very much familiar with. They are unicellular organisms with a much simpler cell structure than other organisms. A key difference between bacteria and other biological organisms is that they have no membrane bound organelles and a lack of nucleus. The genetic material of bacteria is contained in a single loop of deoxyribonucleic acid. Notably, some bacteria also have an extra circle of genetic material which is known as the plasmid. The plasmid is important for the bacteria that contain it, as it contains genes that confer an advantage such as antibiotic resistance over other species. Bacteria are classified into different categories depending upon their different shape. These include rod shaped bacteria, which is known as bacilli, spherical shaped bacteria, cocci, comma shaped bacteria, vibrio, spiral shaped bacteria, spirilla, and screw shaped bacteria, that is Spiroptia. Bacteria can exist as single cells, in form of pairs, in form of chains, or in clusters. Bacteria can be found in every habitat on earth, from soil and the ocean to Arctic snow. Bacteria also live inside the body, where they can provide or they function importantly as it is evident by the gastrointestinal microflora. Bacteria also plays an important role in several critical environmental processes such as nitrogen cycle, whereas some bacteria are involved in food production processes also, whereas others are pathogenic and have caused epidemics and pandemics over the course of human history. Moving on to the next, that is archaea. Archaea is single-celled organism that form the third domain of life. While these organisms are evolutionarily distinct from bacteria, they share several similarities to bacteria. There are many biological differences between bacteria and archaea, which includes a lack of peptidoglycan in the cell wall, as well as the presence of phytanyl instead of fatty acid on the cell membrane. The cell membrane of bacteria is always a lipid bilayer, whereas in case of archaea, it can sometimes be a monolayer. Archaea also contains distinctive translation ribonucleic acid and ribosomal RNAs. Archaea are obligate anaerobes that live in low oxygen environments such as water or soil. Some examples of archaea include Aeropyrum premix, Ignispharia aggregans, and Metallosphaera sedula. Archaea, just like bacteria, can survive in some of the most inhospitable environments on earth, including salt deposits, deep sea thermal vents, and in hot springs. These are known as extremophiles. Moving on to the next section of microorganisms, that is protozoa. They are single-celled eukaryotic organisms that belong to the kingdom Protista. These organisms are often considered to be more complex than bacteria and archaea. The reproduction of protozoa is asexual and achieved by the process of budding, fission, or zygogony. However, some protozoa are capable of sexual reproduction as well. A key difference between protozoa and bacteria or archaea is the presence of a nucleus. Protozoa are motile and capable of moving by cilia, flagella, or amoeboid movement. Amoeboid movement is achieved through the use of pseudopodia, which are temporary protrusions of a cell. Protozoa can reside in a wide range of moist habitats such as soil, marine environments, and fresh water. Some examples of protozoan species include amoeba and paramecium. Although very few protozoa can cause disease, but some are known to be parasitic in nature. Parasitic disease causing protozoa include Plasmodium, which is the organism that causes malaria. Moving on to the next microorganism is fungi. The group of eukaryotic organisms, which is known as fungi, includes mushroom, yeast and molds. These organisms can either be unicellular or multicellular and can range from micro to macroscopic in size. Fungi do not contain chlorophyll and instead absorb nutrients from their surrounding environment. Fungi are classified as microorganisms, yeast which is unicellular, whereas molds which is multicellular and produce microscopic filamentous structures. Some fungi are pathogenic, whereas others are non-pathogenic. That is, they are beneficial and can be used for various medical or fermentation purposes. Moving on to the algae, they are eukaryotes that, like plants, use chlorophyll to photosynthesize and have rigid cell walls. Occurring in moist environments like soil and aquatic environments, algae may be microscopic and unicellular or can be multicellular and large. In fact, some algae species may grow up to 400 feet in length. Multicellular algae can occur in a variety of forms and degrees of complexity. Some form colonies, which can be simple aggregates of cells or contain specialized cell types much like higher forms of life. Moving on to the next is slime molds. Slime molds are enigmatic, both taxonomically and biologically. These organisms are neither protozoa nor fungi, although they share common features of both these organisms at various stages in their life cycle. Slime molds can also be protozoan like during their growth stage, as they lack cell walls and exhibit amoeboid movement. Comparatively, during their propagation stages, slime molds form fruiting bodies and sporangia like typical fungi. The two groups of slime molds are the acellular and cellular slime molds. Next is lichen. Lichens are symbiotic organisms that consist of a photosynthetic microbe like a cyanobacterium or algae, which is intimately associated with a fungus. The bacteria of lichen provide nutrients for the fungus, whereas the fungus provides protective cover for the microorganism, which proves mutually benefit for both. The typical structure of a lichen is a top layer of fungal mycelium, the middle one is where the microbe lives, and bottom layer of mycelium. One of the ecologically important roles of lichen is its ability to turn rock into soil. Next is virus. Viruses are one of the main pathogenic agents that have been responsible for an enormous number of different diseases in humans, plants, animals, and even in some of the microorganisms. These organisms are much simpler in structure as compared to other organisms, as they have no actual cells or organelles. Virus can only replicate within a host cell. When not residing within the host, viruses exist as virions. Virions have a simple structure that consists of a genetic material, a protein coat which is known as capsid, and in some cases an envelope of lipid. Virions are the most numerous biological entities on earth and mutate much more rapidly as compared to bacteria. The key difference between viruses and the other type of microbe discussed here, aside from prions, is that scientists disagree as to whether viruses are technically alive. This classification depends on one's definition of life. While there is no distinct phylogeny yet to be identified, viruses exhibit functions that are essential for life. Some can even form protective structures for their survival outside the host. Next is prions. They are much more simpler than virus and thus much smaller in size. These organisms are obligate parasite that have been found to survive for up to 2 years in the environment. They possess no genetic material and are instead self-perpetuating proteins. These organisms have been implicated as the cause of various diseases such as bovine spongiform encephalopathy and Creutzfeldt-Jakob disease. Next is helminths. They are basically multicellular parasitic worms with complex life cycle, which is responsible for causing disease in humans, animals, impacting health and agriculture. Examples of various helminths include roundworms, tapeworms, flukes and many more. Biodiversity is basically one of the earth's greatest treasures. Microbes, despite their small size, have a huge impact on our lives. Therefore, understanding their roles in the environment is important to the maintenance of our planet and preserving the diversity of plants and animals. Microorganisms have the largest genetic diversity on earth. Billions of species of bacteria are suspected to exist. However, only 1 to 5% or such similar species are characterized in real. Microorganisms, being the pioneer colonizers of this planet and the masters of the biosphere as they considered by some, are ubiquitous and can exist in most inhospitable habitats with extreme temperature, pH, water and salt stress. Microbes are of tremendous importance to man. Worldwide, the economic value of microorganism is estimated to be at least many tens of billions of dollars annually. The microbial application for economic or industrial purposes has advanced considerably with fast paced developments in industrial microbiology and biotechnology. Microbial diversity has provided invaluable service to mankind since the ancient time. Despite the fact that microbes were yet not been discovered in ancient times, people relied on microbes to produce different types of food. Vinegar, yogurt and many types of cheese were the important examples. Almost all the antibiotics have their origins in the metabolites of soil bacteria, especially within a family known as Actinomycetes. Microbes are also used to produce vaccine, providing humans with immunity against several lethal diseases such as measles and smallpox. Many diseases can be treated with microbial compounds such as diabetes, cancer, cystic fibrosis, growth hormone deficiency and hepatitis B. In addition, microorganisms are also factories for industrial enzyme production, which are used to enhance detergents, to clean up toxic waste, to replace chemicals in paper and pulp processing and for oil extraction. Furthermore, microorganisms and their enzyme systems are responsible for the degradation of organic matter. Currently, wastewater treatment uses microbes to decompose organic matter in sewage. Microbes can also be used to create biofuels like biogas or bioethanol. While microbial communities are potentially affected by natural and anthropogenic activities such as agriculture, use of pesticide, pollution and urban development, it is yet not known how changes in microbial diversity can influence the ecosystem. Dry lands have been neglected in both conservation and sustainable use, which, despite the fact that deserts are a fragile ecosystem, has to be handled very carefully. Knowledge of the composition of bacterial communities and how these communities are affected by landscape sustainability measures will find wider application in landscape sustainability programs and contribute to future global policies. There have been many different techniques for studying microbes. Traditionally, we know that microbes were discovered in mid 1600s just after the invention of microscope. So, they were largely ignored until the late 1800s, where people began to realize that microbes actually cause disease. Once they understood the significance of microbes, researchers began developing techniques for isolating them and growing them in laboratory. They used microscopy to study their different physical forms and culture techniques to categorize microbes. Microscopy and culture techniques have taught us a lot and they are still used today, but they provide a limited view of the microbial world. Many microbes look similar under a microscope and many do not grow outside their natural habitat. Thus, early microbiologists were only able to study a smaller number of microbes, those that grew in the laboratory. It was not until DNA sequencing became available in the 1990s that these limitations were slowly overcome. Today, researchers study microbes by analyzing their DNA, most commonly the gene that codes for the small subunit of the ribosome, that is 16S rRNA gene. The ribosome, we all know, is an essential piece of the cell's protein making machinery. All bacteria have 16S rRNA gene, but the exact DNA sequence is unique to each species. So, it is basically used as a type of molecular fingerprint. So, when scientists isolate a microbe, they sequence this 16S rRNA gene and compare it to the already available known sequences using a database. If they find a match, they can identify the species. If they do not find a match, it is believed that they have discovered a new species. Ribosomal RNA studies have expanded our view of the microbial world and revealed evolutionary relationships between species. Closely related organisms have more similarities in their rRNA gene than distantly related organisms do. Genetic data has been used to construct family trees that show the evolutionary relationship among microbes as well as other organisms. rRNA analysis also led to the discovery of a third domain of life called archaea, which is now completely distinguished from bacteria and eukarya. Through rRNA analysis, we have learned that the number of microbial species on this planet is much larger than we thought. The new science of metagenomics, where the microorganisms are studied in context of their natural habitat. All the genes in a microbial community are collectively referred to as metagenome. The latest DNA sequencing technology is taking genetic studies a step further. Researchers can now sequence a metagenome or the complete genome of every microbe in an environment sample. Amazingly, more than 10,000 microbial genomes can be sequenced in a single experiment. Researchers cannot tell actually which genes belong to which microbe, but by analyzing the genes collectively, they can learn what the microbes are doing. This method is significant because even though most microbes are single cell organisms, they live and work in diverse communities. They depend on one another and often cannot live on their own. For example, the breakdown of products of one microbe may be food or another. By sequencing all the genes in a microbial community, the metagenome researchers can identify new species and study genes, which is important for the survival of a community. Already, microbial communities are proving to be a reservoir of useful biochemicals. We hope to learn from these microbial chemists that how to make new medicines, produce biofuels and clean up polluted soil, air and water. Now, let us look into some of the application of microbial diversity. The application is vast, just like the microbial the diversity is vast and span numerous fields. Environmental and ecological application, which includes bioremediation. Microorganisms can break down pollutants in soil and water, helping to clean up oil spills, heavy metals, pesticides and other contaminants. For nutrient cycling, microbes play a crucial role in nitrogen fixation, decomposition of organic matter and other processes essential for ecosystem health. For maintaining or improving the quality of soil, microbial diversity plays a very important role. It contributes to the plant growth, disease resistance and maintenance of soil structure. Moving on to the application in case of agriculture sector. For plant growth promotion, certain bacteria and fungi enhance plant growth by nitrogen fixation process, solubilizing phosphates and producing growth hormones. Next application involves as a biocontrol agent. Beneficial microbes can control plant pathogen and pest, reducing the need for application of chemical pesticide. As a composting process also, microorganisms decompose organic waste into nutrient rich compost, enhancing the fertility of soil. Moving on into the industrial application, the first is biotechnology. So, microbes are used in the production of various elements such as enzymes, antibiotics, biofuels and other bioproducts. In fermentation, microbes are essential for producing foods and beverages like bread, cheese, yogurt, beer, wine and many others. In case of bioplastic, some microbes can be used to produce biodegradable plastic, which offers a sustainable alternative to conventional plastics being used in the market. Next is medical and pharmaceutical application. Many antibiotics are derived from microorganisms. Continuous exploration of microbial diversity has led to the discovery of new antibiotics. Beneficial bacteria can also improve the gut health and are used in development of probiotic supplements as well as functional foods. Microbial diversity has also played, as mentioned earlier, a very important role in development of many important vaccines. For genetic engineering and biotechnological research, microbial models like E. coli and yeast are essential for genetic studies and biotechnology research. Engineering microbes to produce novel compounds, biofuels or to perform specific biochemical reaction, for this also microbial diversity plays an important role. Using CRISPR technology, which originates from the microbial immune system, they are used for precise genome editing in various organisms. For maintaining the diversity and environmental monitoring, certain microbes can serve as indicators of environmental health and pollution level. Understanding microbial diversity helps in conserving overall biodiversity and functions of ecosystem. In food industry also, for food preservation and food safety, microbial diversity, which encompasses bacteria and certain fungi and other organisms, they help in increasing the shelf life of food products. Detection and control of pathogenic microbes ensure food safety and quality. In conclusion, microbial diversity is a cornerstone of life on earth, which plays a vital roles across ecosystem, agriculture, industry and medicine. The vast array of microorganisms support nutrient cycling, environmental health and plant growth, which contributes significantly to ecological stability and agricultural productivity. In industry, as we saw the previous application, microbes are indispensable tools for biotechnological application, which includes enzyme production, fermentation process and bioplastic development. Medicinal and pharmaceutical advancements such as antibiotic, probiotic, vaccine heavily rely on these diverse capabilities of microbe. Furthermore, microbial diversity is fundamental in genetic research and synthetic biology, which drives innovation, new technologies. Understanding and preserving microbial diversity is crucial for sustainable development, environmental conservation and human health. Continued exploration and application of this diversity offers solution to many of the world's pressing challenges, which includes pollution remediation to food security, disease prevention and many more. Embracing the potential of microbial diversity will enable us to harness its benefits for a healthier and more sustainable future. With this, I hope that you have gained some knowledge about microbial diversity. The next topic which will be discussed in this SWAYAM course is about pathogens. Different pathogens, their type, some examples will be discussed, the pathogenicity level and how they are harmful for the environment, which includes humans, animals and all the living beings will be discussed. With this, thank you very much.